to have you join us for the Eminent Scientist Dialogue Series, which entitled The Relevance for the Exposome on Host Microbiome in a One-Net Paradigm that promises to be both enlightening and inspiring. It is an honor to welcome our guest, Dr. Bharat Prithviraj, Senior Microbiome Scientist, Ricket Health, USA. Dr. S.R. Rao, Vice President, Research, Innovation and Development, Sri Balaji Vidyapith, Professor Ananthakrishnan, Dean of Faculty and Professor of Eminence, Department of Surgery, MGM CRI, Sri Balaji Vidyapith. Now I invite our Honorable Vice President, Dr. S.R. Rao, to deliver the welcome address and to elaborate the highlights of the program.
Good morning to all of you. In fact, it's a series of the kind which we initiated on the name of Eminent Scientist Dialogues. Then many people understand eminent means he should be very old. He should have 40, 60 years of experience. Eminence means scientific eminence. How many papers he has published? What is his presence in the subject? So, when the proposal to have this meeting from our Dr. Damodaran, he brought Dr. Bharat Prithviraj biodata and said, "Sir, can we have him under the Eminent Scientist Dialogue?" Then for a while I was apprehensive in the first page. And then as I went, I thought, yes, why not? So it's very important today. Microbiome, since last 10, 15 years, is a buzzword. And several people in the country had lots of projects, 3 years, 5 years projects. And sometimes they generated the data and they left it there. We were just discussing before we came here. I like the word he said, unless our work is contextual and it has many meaning for the public health or the patient care, or how this bench knowledge can be translated into bedside, there is no point in going on accumulating sequence data after sequence data. So what we did was to examine this one. People from MGMRI, particularly Dr. Ramya and Damodaran and other people joined together. The first thing we wanted to take a stock where India is. So we made a database of microbiomes of skin and gut microbiome. So all organs, whatever is been published. And you can do a searchable database. Initially, we had 10,000 records. It was got released by Director of National Center for Biological Sciences, Dr. Shashidhara, on MGMRI Day of 12th December last year. And then we added some more data. Now it's around 16,000. 16,000 records are there. It's a huge data of sequencing, everything one can access. What we notice is, in many of these studies, the word contextual is missing. We are just accumulating the data, and then data is, now it is up to why this database was created, was at least those people who are interested to make it contextual need not to again go and source those organisms. They will have a background at one shot. So that is one great achievement in microbiome. I congratulate Dr. Damodaran and also Ramya, who is a bioinformatician, and others who have been in this gang to make it happen. Now, as far as microbiome is concerned, there is not a single lab in the country, as I know, because of my own past experience in the funding agency. I can easily say a statement where people are linking this bench to bedside story. This is where we want to be having advantage. Damodaran and he's working with dental science people on oral microbiome, both in terms of oral cancer and otherwise the oral microbiome as such. And he's also collaborating with our hospital on various aspects of gut microbiome. So it is a good opportunity that you are here and you know this place very well. And it's a private university. People cannot go on funding our research without any end results. So I would be very happy that this initiative and you are coming can bring more fruits of collaboration. And I'll be very happy to, or the university will also be very happy to facilitate any kind of agreement or MOU with your organization or any other initiative so that we are in the loop and we do what is very important for microbiome research. And I have been talking to you in the few minutes ago. So that is the main crux of calling people from all over the country and the world. And the second important thing is that MGMRI is a budding institute. We have people with 16 different disciplines. So I told you, somebody specialized in microbiome, somebody in genomics, bioinformatics, stem cells, like that. So it's supposed to have a very good collaborative effort. We're not duplicating the expertise. So under one roof, when you start attempting microbiome collaboration, we can address many issues. So therefore, I welcome you with these words, and we are very happy to have you here. And after my interaction in the morning, I am sure that we are going to be benefited a lot, not only by your talk, but also in future. Thank you very much.
Thank you.
Thank you, sir, for that insightful address. May I request Dr. S.R. Rao to felicitate our chief guest with a bouquet?
Thank you. Thank you.
I now request Professor Ananthakrishnan to felicitate our chief guest with the memento.
Thank you. Pleasure to meet you.
Thank you, sir. Now I invite Dr. R. Damodaran, Assistant Professor, MGMARA, to introduce our chief guest.
Good morning, one and all. So it's my pleasure to welcome Bharat Prithviraj. He is a senior scientist in Reckitt Biotech. So basically, he is from Pondicherry and he did his master's in Pondicherry University in School of Life Sciences and a PhD in MS Swaminathan Research Foundation, Chennai. And he is a famous microbiome scientist. He has a vast list of research experience starting from, he started his research career in Center for Biotechnology, Anna University, followed by he worked as a senior research fellow at MS Swaminathan Research Foundation and he did his PhD there actually. He was fortunate to have Dr. MS Swaminathan as his PhD, like co-guide. And he received International Scholars Award during his PhD period. And after his PhD, he did a lot of postdocs. It started from, he worked as a senior scientist in Anthem Bioscience, Bangalore, and research associate at University of Colorado, USA, and postdoctoral research fellow in Noble Research Institute, LLC, and postdoc in Mason's microbiology lab at the in School of Cornell Medical School. And he also adjunct assistant professor in City University of New York. And later he worked as a senior scientist and prime discover microbiology lead in RR Biosciences, and consultant R&D at Pitti Biomics, New York, and senior scientist. Currently, he is working as a senior microbiome scientist in Rickett Health, actually. He is an early member of Earth Microbiome Project, and he worked in many different projects in the field of microbiome, microbiomics, actually. He has well experience in microbiome research over the decades, actually. So his experience include metagenomics, metabolomics, proteomics, probiotics, skin and gut microbiome, and he is proficient in high throughput sequencing and microbiome informatics. He developed a lot of protocols in the field of microbiome and metagenomics, actually. So with his vast experience, I'm glad to invite Dr. Bharat Prithviraj to deliver a talk. So I also welcome all the, good morning, sir. I welcome our Honorable Vice Chancellor who joined online to witness this eminent scientist dialogue. Sir. Good morning, sir. Thank you.
Good morning, one and all. Thank you so much for the invitation, Dr. Rao, Professor Ananthakrishnan, and students, faculty. It's a pleasure being back here in my hometown. And incidentally, it's my first official public interaction talk in Pondicherry. Although I've been in other places, but I did have interaction in Jipmer maybe before COVID, but that was more of a smaller scale, but it's nostalgic and good to be back here. Hopefully the online audience can hear me. And trying to get my screen up. Yeah, okay, thanks. Thanks for the introduction, Dr. Damodaran, and it's a pleasure being here. So as mentioned, so I'm part of a company called Reckitt Health. They used to be known as Reckitt Benckiser. And if people don't know of this company, you're probably very familiar with the products that the company makes. And I can name one, which is very familiar, called Dettol. And any one of us growing up in India would have used these products. So I'm part of the global R&D team called Science Platforms. This team was essentially assembled post-COVID, where as an organization that's basically a fast-moving consumer goods company, FMCG is the operating acronym for it, saw the need to engage with the consumers on research areas that are relevant to day-to-day, well-being. And given that there's so much of insight coming out of the field of the microbiome in the last decade or so, there is a very certain need to contextualize the information that you're generating from each of these data-centric efforts and present that in a very lucid manner such that the consumers can be aware of what it means and what is the relevance of it. So besides Reckitt, I do have affiliations. Again, that's my commercial affiliation. I do have nonprofit affiliations, starting with the MetaSub Consortium. And most of my talk is going to focus on some of the efforts being done over the years for that project, as well as I do advise initiatives such as the New York Urban Soils Initiative, which actually works with the city of New York, which is in fact an environmental stewardship initiative, as well as a Small World Initiative, which is actually catered towards school students to instill in them the interest towards understanding the microbiome, as well as areas such as AMR, because that needs to be instilled at a very early age, and that understanding of why care about the environment that we live in and how that has ramifications for health as well, right? So I'll begin my talk with a dedication to a guru. And as mentioned, I did my PhD at MS Swaminathan Research Foundation, and Professor MS Swaminathan was my doctoral committee member, and he was recently conferred with the Bharat Ratna. And I do want to pay an homage that we remain inspired by the vision of his vision to democratize science, expand horizons of using biodiversity and conservation, creating a relevance for global challenges, one of them in climate change included, and also embed translation of next generation biotechnology tools that can help augment the deliverables for grassroots initiatives. Starting off with that, yeah, let's get into the crux of these topics, right? So I want to reflect back on a study done by Bach et al. In 2002 from NEJM, where they showed the inverse relationship between infectious diseases and immune disorders over the years. And if you can notice that there's an uptick in terms of the immune disorders over the years, given the time span that we are looking at from 1950 to 2000, and basically an inverse response for these infectious diseases. And inherently, there seems to be a trigger factor associated with the microbiome, which brings about these challenges. On top of it is a disease called Crohn's, which actually has tremendous ramifications in terms of how it's become more pervasive, not only to the Western countries, but also to the developing nations as well. This is associated with lifestyle changes, dietary changes as well, that is compounding the effect of how intense these representations are happening. From here, I just want to reflect back on the microbiome. We always keep talking about how important this is, this being the second genome, this being the most dominant spatial relevance in the human body. It's also a good opportunity for us to step back and examine the relevance between microbial cells in the body and how spatially relevant they become. So here on the left is a figure which actually came out of a paper in PLOS Biology, where they computed or basically make a representation of the body mass cellular hierarchy. And here in color coding are the different cell types. And you can see that in total human cell count, the erythrocytes make up the majority of the cellular constituents in the body. And the other regions are pretty much, less in terms of representation spatially. So if you compute that by cells, if you think of coded in red, 25 trillion cells represented by erythrocytes, you have 38 trillion cells from bacteria that are represented in the human body. So the disparity is really like obvious there, that you have the human genome, which again is the most popular data point that was generated over the years, but then the relevance, the spatial relevance for the microbiome becomes even more important, given that you have cellular constituents that are distributed based off of their mass. And here you only have 0.2 kilograms of the microbes represented here, whereas you have, again, if you think about muscular cells, 20 kilograms in terms of mass represented as an individual. It's kind of important and helpful to put in perspective how much of impact this can create, even though in terms of the mass representation, it is diminished when you compare it to the other cells of the human body. So I'll take a step back and kind of go into a little bit of a basic biology sense here. So I want to talk about selection pressure and macroevolution. So given that we study biology, a lot of the insights that we get from the information generated through biological hypothesis makes sense only in the context of evolution. And as Darwin put it there, you have species that are formed over several years, several decades, several generations, several timelines. But the adaptation of these species is driven by selection pressures that are instilled at every given point in time, every given epoch. And that's going to determine how these species survive and become pervasive through how they represent themselves in different hosts. In this case, we're talking about human hosts. And how did we get here? How do we get to an understanding where you have a unique representation for species, either host-associated or free-living in environments? I just want to quote Professor Norman Pace, who I had a chance to work with during my time as a postdoc at UC Boulder. And Norman Pace was the pioneer to bring in environmental metagenomics in terms of democratizing it, the approach of utilizing an ability for us to study a large number of microbial populations using consensus genes, in this case, 16S ribosomal RNA, was pioneered by Norm. And he actually did his training under Professor Carl Weiss, who was actually the person who discovered archaea. There are the transition groups of microbes between eukaryotes and bacteria. And archaea, as we know, is a close relative of eukaryotes. In this case, in later on sequencing efforts, it's been come to light that they're literally overlapping sequences with the eukaryotes when you look at the archaea more closely. And it's tremendous to think of the fingerprinting ability that the 16S ribosomal RNA brings in terms of helping us understand the identity across organisms, as well as the variability between organisms. It's tremendous power with a very frugal effort that sort of became the starting point for some of the microbiome initiatives in its infancy. And you have this ability to have culture-independent investigation of microbes, and all of cellular life has a shared history in terms of evolution, in terms of how it responds to selection pressure, how it creates a unique differentiation for itself. And we can have the ability to utilize sequences that can act as a genetic fingerprint, in this case, very specifically a microbiome fingerprint, which is very unique to each individual in terms of how personalized it can become. I do quote this famous scientist, Dobzhansky, nothing in biology makes sense except in the light of evolution, which is so powerful. As we know, culturing microbes is extremely hard. And you basically have less than 13% of this bacterial genera. In this case, I'm using example Bacillus anthracis in culture. And many of these approaches for understanding organisms have stemmed from culture-independent methods. And a lot of the metrics for computing diversity is derivative of these data points that you've generated and accumulated over periods of time. So much so that now you have a new tree of life, starting with the one that I just showed, which was 16S alone. Here you have a tree of life that's been computed using assemblies of genome sequencing. In this case, these are metagenome assemblies that have been stitched together to provide information on the gaps that are existing between these domains that I talked about, bacteria, archaea, and eukaryotes. And this work was done by Professor Jill Banfield's lab in UC Berkeley, where they were able to show that there is a superphylum, which is represented in the archaean groups, that has an outlier that feeds into eukaryotes. So you can call the archaeans as the missing link between humans and microbes that make them even more and more interesting. I have a quick question for the audience. Can you name an archaea that you would find in your own gut? There's a group called Methanobrevibacter, Smithii. So usually when you represent that in the data, people think that's bacteria, but these are archaeans that are having a very niche role to play in the human gut. So they are very environment specific, known to be very prominent in hypersaline environments, but you still find them in the human gut. So it's an interesting group in that sense. So yeah, Hugh et al. Published in 2016. In fact, this was from Professor Jill Banfield's group. A lot of the work that they initially did was on soil microbes. They used to study dark matter. When you have deep soils, they are extremely diverse and they have very, very unique constituents. And from there was how these insights was born in terms of how you can unravel sequence information and stitch together information that you can put in the context of understanding these communities. So if we think of, again, the evolution piece of it, we talk about phylogenetic patterns of how the interrelationships between organisms is maintained. It also becomes important and pivotal to understand them in the context of time, because selection pressure has a direct relevance to time. Over periods of generations, you have these adaptations happening, and random mutations are continuously occurring at different timescales. And when you phylogenetically assemble these sequences and you want to understand diversification between microbial groups, you have a phylogenetic tree assembled and the sequence information computed in the context of the phylogeny, which means you are representing the relationship between groups of organisms using their phylogenetic interrelationships, or the distance of how they are separating from each other based on the phylogenetic tree of where they are constitutively represented. I'm bringing this information because it becomes relevant when we ask questions on how, as a host ecosystem, we respond to the environments that we live in. And here, there's a concept of the hologenome and the holobiont. Basically, this distinction is important for us to know that as a host, we have inside of us different organisms that have their own properties of symbiosis. They associate or disassociate with organisms, and they have their own methods of coping with selective pressures inside our own host system. So that is represented in a phylogenetic signature that you can also unravel using computing methodologies. And to put it in context, you want to understand how these selection pressures that are creating disturbances within, say, our own gut or an ecosystem, which is more compartmentalized response or creates a response for microbial groups. So you can have different types of these responses. One is you have organisms in a community that are together that can present resistance to any disturbance that you're bringing on. For instance, you can think about consuming antibiotics as a factor that creates disturbance effect on a community of microbes that you have. And you have going to have a set of organisms that are resisting that interaction. So they're going to push back, and they are not going to change whatever you do, whatever you introduce. But then you have another cohort of organisms that show a sense of resilience. It's actually one of my favorite traits in microbes. What happens is, initially they're vulnerable, but then they push back and then come back to their original composition. It's like, I want to take an example of tennis. One of my favorite players is Rafa Nadal. So he's one of the most resilient players of all time. You can find him in losing situations and then he just finds a way to come back out and win, right? So that's the best trait of succession or even survival. So I'm just using a sport analogy here, but I think these are one of my favorite groups of organisms. And also there's also functional redundancy, which is you have a disturbance effect meted out to a cohort of communities. And then the groups of these organisms, they change. And then what happens is the change is not so deep in an extent that it alters how that original community was behaving. It basically becomes redundant. So there are these three different tiers of how organisms respond. This becomes important, especially from an environment, also from a compartmentalized environment's perspective, to understand phenomena such as resistance. And the reason I want to bring that up is each of these organisms present a property or a trait on how they respond to their original community structure and how they reflect back to the resilient trait or the resistant trait or a vulnerable or a redundant trait through their metabolic signatures. So Evelyn Hutchinson is a ecologist. Again, he is someone who used to study macroecology, organisms in the environment, and also understand why there are certain organisms preferring to live in very specific ecosystems. And how do you even understand their response to different environments where they're introduced to or where they're exposed to? So that initial theory around niche theory is habitat niche or preferences for living in certain regions. I took an example of archaea. Methanobrevibacter has its own unique niche, and that niche is influenced by the functional repertoire or the metabolic signatures that are presented by a region in the gut that it prefers to occupy, and hence it co-localizes itself in that region. So it's staggering to think of some of these concepts in macroecology translating into microecology or in the microbial evolution realm, which is where a lot of the beginning stages of the microbiome was very, very helpful. So much so that when I started my postdoc in Professor Rob Knight's group at UC Boulder, 2010, he came up with this idea of this outlandish project called the Earth Microbiome, which is to catalog the microbial niches of organisms all around the planet. And we were like, this is daunting. And for me, embarking on a first postdoc project, I was like, where am I headed, right? Am I going to see the end of the tunnel? But it was a collaborative effort, 150 institutions around the world and 264 collaborators together pieced in information which involved end-to-end sample preparation guidelines, replicating the guidelines for sequencing as well as data analysis. It became the backbone of a citizen science effort in the US, later on called as the American Gut, because a lot of the protocols that we use to sample or to process samples were utilized for creating that metric of replicability. The project started in late 2010, early 2011, and finally got published in Nature in 2017. So it was like a long 5-6 year wait for me to get to that stage. But I think the learnings were tremendous in terms of, I think Dr. Rao mentioned contextuality. We've been talking about it, right? So we wanted to understand, yes, you have select environments where you have groups of organisms that are pervasive and present. Why are they present? What is influencing their presence? What can also modulate their distribution? So those are the questions that we were asking at the time. And I think this was an open source data set at that time, was having 27,000 samples, specifically looking at 16S microbiome information from bacterial communities. And we were able to catalog different sample types from different environments, saline environments, water, aquatic samples, sediment samples, free living samples from non-saline environments, water, sediments, soils, host associated, except human. We looked at animal guts, plant associated communities, and how they distribute at different spatial relevance in terms of their diversity and how they're different or similar to each other is those questions that we were asking. So between a host associated gut system, non-human in this case, and the environments, are there any similarities? Or are we even connected to the soil that we are living in proximity to, right? So those were again, those further questions that we wanted to answer. So this was the backbone for that. And we also showed that these abundance metrics are driven by two primary categories. So if you can see here in host-associated groups, you have the plant rhizosphere, which is where a lot of the biological activity happens for a plant to survive in a soil or in a farm, right? Is actually acting as the most diverse in host-associated systems. And the same thing, obviously, with soils. is the most diverse in free-living ecosystems, as you can see, represented by their overall phylogenetic diversity. So this is actually, again, in that paper that was published. And also, we could derive patterns out of the phylogenetic relatedness between these organisms. So what you're seeing here is a principal component ordination plot. It's basically a data reduction technique where what you want to do is, out of those 27,000 samples that I mentioned, you're computing the phylogenetic relationship between each of these sample data points in a given habitat that you've studied them and how interrelated they are between each other, right? So in red, you have samples that are coming from soils. And then here you have in green things that are associated with plant surface. And you can see the plant rhizosphere interspersed with soil because they are most diverse. And you can also look at communities such as aerosol, which is non-saline, but aerosol is constitutive of microbes you find in the air, in the resuspended air. They are all interrelated in certain extent within the habitats that you're studying them in. Why this becomes important? Because across environments, there is key signature. If you want to understand this interrelationship between species, host-associated, and free-living environments, it's important to know the interconnectedness between the human hosts as well as the environments we live in, driven by the animals we coexist with, the environmental chemicals and exposures that we have through diets, through day-to-day exposures of utilizing day-to-day products. And also the manifestation of disease and how that is influenced by lifestyle changes. Again, the more and more developed or the more and more westernized we become, all of these influences are going to trigger responses in how you recuperate to the onset of infection or how your immune evasion mechanisms are stimulated. So that becomes a contextual phase for that. So using the Earth's microbiome, one other question that we were able to ask was, how are the human and environmental microbes related to each other? So there's a paper published by one of my former colleagues, Daniel McDonald, in Systems, where they computed the phylogenetic relationship between data coming out of the Earth Microbiome Project, EMP, and data coming out of the American Gut Project, which is primarily host-associated, just human gut-associated. And you can see that in terms of dissimilarity, you have two ends of how these samples are separated out. But at the same time, you have an overlapping area where you have commonalities between soils and human gut. Furthermore, if you look at this phylogenetic tree below here, you have samples coded by the color here. Red represents the HMP, the Human Microbiome Project. In blue, you have the American Gut Project. And then in gray or white, you have the Earth Microbiome Project. There are specific pockets of these phylogenetic trees that are represented by phyla, such as firmicutes, which are extremely abundant in the human gut, by the way. And also groups in Bacteroides that are interspersed with the EMP samples. So which gives us the relevance of exposure or intake of organisms that again translate to equation where you can see the industrialization effect and the loss of communities, which has resulted in very sparse representation of environmental organisms in the human gut. Usually you would typically see more of a diversity in this, as you can see in studies where they track down hunter-gatherer communities that have even more environmental microbes in their human gut. And you see that factor depreciating in the industrialized populations. But by the way, these industrialized populations are from the Western world, which is again another point of a gap in terms of data equity, in terms of health equity as well, which we need to have initiatives floating around, especially to target or also access data from the communities that are in regions like South Asia, which is more diverse and more populated. So once we understand that there is a depreciation of microbial diversity overall, it brings us to the question of how does that lack of diversity impact us overall, right? So it's raised the question of resistome. In this case, when I talk about resistome, it's the genetic makeup of the microbial genetic makeup of organisms that are relevant in the human gut right from the time we are born. So the spatial relevance of the lack of diversity is impacting us in specific ways where from the time we are born, there is a selection pressure instilled in a baby itself. And selection pressures can come from the mode you're delivered from the ingestion of antibiotics during the point of birth, from the way you're hospitalized. For instance, if you have a premature infant, there's going to be extended hospitalization period. And then if you have, again, as a mother, you have a maternal infection onset, early onset before pregnancy, that can also have these selection pressures instilled. So you could reflect back to that flow chart I showed you about, how disturbance can influence the resistance and resilience and the redundancy, right? So if you reflect back on this, this also will translate into some of these patterns that we see. And we talk about the resistance. Obviously, we want to talk about the elephant in the room, which is resistance, antibiotic resistance, which emerged as the largest public health threat post-COVID or even pre-COVID. And basically, the number of deaths over the years are projected to be in the order of 10 million in 2050. And these numbers are staggeringly, throwing us off proportion in terms of estimates. It's increasing and changing over a period of time. And it's a topic that needs to come into the mainstream dialogue, especially as we talk about research, we talk about problems in the areas of the medical community, working in, collaboration with the scientists who have the ability, as well as epidemiologists, public health experts, together to kind of address this global challenge, which is now named as the silent pandemic. This is a map of the global prevalence of AMR in animal reservoirs. And if you go to resistancebank.org, they actually updated a recent publication with the spike in the national level percentage of ingredients that have greater than 50% of resistance exhibited. India is sort of in the, maybe like, I want to say 12th place here or somewhere around that, between the top, like kind of like in the top 15, which is something that you don't like or want to preferably see, but that's the reality of the situation. But they compare this prevalence of AMR in animal reservoirs. We talk about chicken, we talk about pork, like pig and cattle. So those are the distributions that were used. And this is a little bit of a narrowed down distribution in what's there in Bharat, like in India, with livestock heads as examples. So you see, in terms of antimicrobial use, you have 5,000 tons per year, which is again increasing in incremental forms, like 5% is going to increase in 2030. And then you have and like basically animal reservoirs. These numbers are going to keep increasing. And this is kind of like the distribution of the percentage of AMR across different geographic regions. So there are interesting patterns that could emerge from here. People will want to ask, what is the frequency of meat consumption in these geographic regions? But I don't want to get into a political conversation with this, but yes, it's good to have it in perspective in terms of utilization of livestock, utilization of, again, you have meat, you have produce. Having guidelines for the guided use of antimicrobials is becoming more and more relevant and more important to have, especially across the stakeholders. And in fact, me being part of an organization that's producing antimicrobials in this case, not antibiotics, but biocides, we also have a stake to play in this endeavor as well. So with that, I just want to go back in time and take an example of an incident that happened in late I think it's 2008-9. There was a study done in Delhi by a research team from the University in Wales, in Cardiff, where they discovered the NDM1, New Delhi metallo-beta-lactamase. Basically, it's a gene that produces a protein, NDM1, that confers resistance in drugs that are carbapenems that are active against antibiotic-resistant organisms. And again, so this particular gene was actually recovered from soil samples in the Arctic several years later. And it became a moot point as to the initial report for this gene came from sewage samples, in this case, water samples from New Delhi. However, the spatial relevance for that discovery was not followed up on in terms of surveillance metrics. So a source to sink dynamic is always required when you want to ask the question, what are the selection pressures that are associated with resistance buildup? And how do we track that back in time? So this basically presented an opportunity for a biosurveillance model and an opportunity to do a geographic association of what is it that presented the selection pressure so much so that this was permeating into the Indian peninsula, right? So I'm taking this as an example because this actually shows the need for a relevant monitoring mechanism and a standard approach where you can quantify and contextualize information in the context of the environment where these organisms are discovered. With the distribution of NDM, one of the things that came up is there are different, again, you have different mutations associated with how this protein modifies its conformation. And this is the surveillance distribution of international dissemination of human cases associated with NDM1, and surveillance of isolates with NDM1 in enterobacterials, very specifically where that's the group that it targets. The first discovery for that was done in a patient who came from Sweden for treatment in India. And then you have, again, Klebsiella pneumoniae, the strain where they were able to discover this beta-lactamase as well as where it was reported from. For some of these surveillance methodologies, what is being used? Laboratory surveillance methods include standard serotyping methodologies where you have diagnostic procedures for clinical samples and you're doing diagnostic tests with cultures, and you would have combinations of, again, typing, subtyping methodologies, as well as pathogen identification using Molotov and biochemical tests. And there's also surveillance using foodborne pathogens, which is actually being done for the food industry, where we want to track food contamination and infection from source, as well as sewage surveillance, for instance. I actually picked up pace because of COVID. A lot of the surveillance was driven by COVID-associated monitoring from sewage samples and showing whether you can make predictions on how it's bound to increase over time, as well as healthcare environment surveillance, which actually becomes very important to know what are the other trigger factors associated with not only the use of antibiotics, but you have other ingredients that you're using as antimicrobials, which include chemicals that are used to control the impact of pathogenic exposures, right? So those are also very important in that sense. There is implication for the human resistome in this. The reason I bring that up is there was a study done in a few years ago, actually 2021, where they looked at 15 volunteers, both male and female, and they isolated samples from different parts of the colon, of the gut, and they wanted to ask the question, what is the concentration of the antimicrobial drug classes in the spatial relevance areas of the gut? Interestingly enough, they found that the AMR classes were very much depreciated in the stool samples, versus they were more prominent in the other regions of the gut, which is actually staggering information, because earlier it was presumed that you can pick up the largest signature for AMR from the gut samples through the stool. But on contrary, this study showed that you have different distributions criteria that can show representation of AMR classes that are diverse and how depleted the stools could become. So between the, again, the lumen and the lumen and the mucosa, again, you're having differences between how the distribution can happen, but it actually raises a question as to, yes, we know that stool is the gold standard for isolating, again, microbiome signatures, but perhaps there are other niches where you need to go and identify and look at the organisms. I bring up the concept of the niche again, because within the niche of the colon, you have different regions like the descending colon or the transverse colon or the ascending colon. You could have selection pressures imparted on the organisms in a different way from how it happens in the rectum. And that can influence functional metabolic signatures that can modulate the growth or the enrichment of AMR classes, which actually needs another focused approach, which is not only looking at the identity, but also understanding the functional repertoire of the organisms that are constituted in the microbiome, which becomes very important because if you think about this, it's also relevant for studies where they show that when people move from a different geographic region to another country, they have a complete difference in their microbiome profile. So Western countries, when you're consuming the diets over there, you have a different influence on how that interacts with your gut at the niche level in a different spatial setting. So that actually creates an impact, creates an opportunity also for us to, in a very specific manner, understand the methodology to track and create the signal for relevance, and also create, say, a point of care. Maybe it could be a point of care diagnostic that is specific to that niche that you want to look into and tap into. What happens again with the resistome is the effect of how overall exposures are going to impact. I talked about moving from one country to another. Again, in terms of environmentally changing your location, that's also going to have an impact on how exposures to other factors that influence the resistance comes about to be. And here I have taken an example, intentionally taken an example of biocidal resistant microbes. And when I talk about biocides, these are chemicals that you would have used during your COVID phase. And you would have maybe started using it then, talk about disinfectants use over and over. So there is also the propensity of microbes to pick up resistance through horizontal gene transfer. So I talked about the example of NDM1. Again, you have these transposons or mobile genetic elements that are there in microbial strains that can shift from generation to generation and bring with it the transfer of that trait information, which gives them the resistance profile that can be transferred, which is also a risk build up over time due to a non-guidance in how often and how long you should use ingredients for germ control, right? So this actually is a responsibility, again, for everybody and organizations that are also producing ingredients for control, for germ control, for germ kill as well. In natural and built environments, I'm taking further into that same example of biocidal resistance, we talked about horizontal gene transfer. How do these happen? I'm taking a very specific example of quaternary monomer compounds. These are compounds that are used for disinfection. And you have transporter genes that are present in microbes, in this case, bacterial groups, that can provide the opportunity for the organisms to pick up resistance and evasive mechanisms to cope with the ingredient that you're using for germ control. And an ability for us to track them down and understand these patterns of response to the ecosystem or the environments that they are part of is becoming more and more relevant and more important as we want to track them down further. Which leads me into a project that I've been associated over the past 8 years called MetaSub, which is associated with metagenomics and meta design of urban biomes, urban environments, as well as subways. So the study started in New York City at Weill Cornell Medical School, where the first effort of its kind was to understand what are the microbes that coexist in built environments where people congregate for different reasons, not for a meeting place. They basically go from place A to B. And New York City, as you know, has the widest or the largest subway network in the world, like compared to London as well. So this was a project that was funded by the Gates Foundation in 2016-17. And the major goals for the project was to understand the spatial environment relevance for metagenomic patterns that are coming out of these built environments, where humans are the ones sources that are transmitting microbes into those environments. And are we able to pick up signatures from antimicrobial resistant traces in these environments? And also identify from the metagenomic information in there, biosynthetic clusters that could give us the opportunity to identify signatures of genes that will help us design the next class of antimicrobials. So that is actually the 3 overarching goals for the project. So the first city-scale metagenome profile was actually published in 2015 from New York City by Chris Mason's group at Weill Cornell Medical School. And in fact, the Wall Street Journal also covered that information because we had a whole wide array of information. This is not a 16S specific effort. This was a shotgun, which is a more deeper way of understanding metagenome information and providing CT scale resolution of how you can find microbes in different parts of the city. In this case, different niches in the city. The reason I call that is New York City is made up of several boroughs, some near the ocean, some near the rivers. So all these environments influence the organisms that you would find in these spatial settings, as well as the tourists that pour into the city every year, literally, from different parts of the world. So that's actually an interesting thing. What did we learn from that, right? We also see the evidence of resistant organisms in these environments. So here's an example of, and Luria brought, you have kanamycin and chloramphenicol, ampicillin, as well as resistant organisms presenting themselves. Basically using a plate-based assay. In this case, we're just looking at phenotype. I know that this is less than 1% of what you can culture, but still you see these patterns that are coming out of the environment. So you see that these resistant organisms are present, and we are coming in contact with them on a day-to-day basis. This is pre-COVID, by the day, but this data. So again, as I said, the Gates Foundation project was to look at the global distribution, epigenetic stratification of AMR, and do it in environments, built environments specifically. What happened with that study was every year, starting in 2016, we call this the Global Sampling Day, which coincides with the Ocean Sampling Day. When it's summer solstice in the middle of June, in 24-hour period, we coordinate with 60 cities around the world. And in 24-hour period, we collect samples in real time and geotag them from different cities around the world. And this we have been doing over the past 8 years before COVID. It started in 2016, as I said. The idea being that we want to collect the contextual information for where these samples are collected from, which location, so that we can go back in time. If we want to go back and look at the same sample, we could do that. We barcode that, and we have an ability to geotag it, as well as to have a provenance where we can trace that sample and where it's coming from, and create these maps of AMR representation in these environments. From the first study, which we published in Cell in 2021, from 60 cities around the world, we had samples collected from the largest metropolitan cities, New York City, London, Rio, Tokyo. And both in Rio and Tokyo, samples were collected during the Olympics, 2016, as well as in 2021. So we have a distribution of organisms which we see that are both novel as well as known. And in them, you have combinations of organisms that are commensal in nature, beneficial, those that form biofilms, those that are non-commensal, and some of those organisms which are those you find in your own gut. And we also made a representation of the maps of AMR drug classes that are represented across continents. And we see a spatial distribution of sample differences between, say, North America versus Asia. So the disparity is different from each other. As well as these patterns emerge in such a way where we are able to map back similarities, which we see in terms of the communities that emerge from built spaces. One of the biggest signature was organisms that are specific to our skin that were more common in these samples from environments that we find. And we also had follow-up studies that were done from this. So we published this paper in, I think, environmental pollution, but we also looked at how the constituents of antimicrobials, in this case, an example being triclosan, is influencing the transcripts per million in terms of reads for AMR classes, right? So you have different classes starting with aminoglycosides and again, fluoroquinolones and so on, but tetracycline, these are all conventional like basically antibiotic classes. But here you have a bigger combination that says unclassified or triclosan. Triclosan, as you know, is a biocide, which is actually now banned, but still it provides an effect on how much of the impact it can create in terms of environmental resistance buildup. And also with these organisms, we find that there are novel categories of microbial taxa that are yet to be unraveled and have these metabolic classes of organisms or gene clusters that are there and provide us an opportunity to mine into for the next generation of antimicrobials, like in this case, type 3 polyketide synthesis, PKS, or known categories of organisms that you could tap into for the next class of antimicrobial or ingredients in terms of drug classes. So this is actually interesting in a way where we are also able to show in terms of global distribution of the urban environmental resistome kind of associates with the socioeconomic and the medical status of these nations. And you can see a big spike in ARG numbers, antibiotic resistance gene numbers in countries like Nigeria, for instance, and also specific categories in select nations. So the biggest frustration that I have right now is on these studies that we have done so far, we don't have any of the India data points. That's going to quickly change. And hopefully we will be having some actionable insights in the next year or so. But we do have this map of between the ratio of samples, the ARG numbers. There is a categorization of environmental signatures for antimicrobial resistance. And at the same time, there is information on categories which are yet to be discovered. Some of them are unclassified, that we are not able to map them back to the data that we are seeing. And this further extends to us asking the question, between these cities, how is the overall environmental influence of the permissive effect of antimicrobial resistance? We find that there are categories that are associated with air pollution, for instance. Particulate matter 2.5 is actually a factor in the environment which constitutes the direct relevance of air quality, and how that influences urban resistome is also something that we have just presented in our publication as well. So this was led by our collaborating team in China. However, it's in collaboration with the Cornell Medical School as well. So I do have provided links to these manuscripts that I'm quoting. And given that we talked about the environment, there's also a very selective niche associated with the air that we breathe. And we have resistome profiles for aerosols, in this case, biological aerosols, where we have patterns of microbes that are distributed differently across cities. So here we compared two big cities in US versus London and then Sweden and South Asia, in this case, Hong Kong. And we find that there are classes of microbials or classes of antibiotics that are distributed differently, and you have specific niche organisms which are commensal in nature. For instance, Curibacterium acnes is something that you find topically growing on your skin. It causes pimples. It's not something that's pathogenic, but you have other organisms that can potentially be triggered to pick up resistance over time. However, there are groups of these commensal organisms that we find in the air. So Professor Mark Hernandez, who also I had an opportunity to do a postdoc with, was part of this study as well. And he's the environmental engineer who is an expert on bioaerosols, where he has designed and invented devices that can help you characterize organisms that you find in the air, and then use that in a very selective way to sequence and understand patterns out of. Leading on from these data points, what I want to come back into is the opportunity that we have here in a level of doing surveillance for AMR with an initiative called MetaSub, where we could bring this particular model to India as a pan-India MetaSub initiative. And we do have samples collected from select cities as well from the 2016 time point. And right now, we want to address this challenge of tracking or surveillance of AMR using the environmental reservoirs as those niches where you can pick up these signals from, and help use that information as a guidance to the public for the impact that this could create, and also engage with the policymakers as well as the governments on, okay, what are the measures that we need to take to control, to be aware of where you have hotspots of resistance built up. So that's actually the motivation behind that. So I launched this way back in 2016 in Hyderabad, before the metro started in Hyderabad. And actually, we got permission from the government there to collect samples. And I want to acknowledge colleagues Narasimha Rao and Someshwar Rao from Osmania University and Pons Bio, where we went into these metro trains, which were fresh off the board, like literally not occupied by humans, to create a baseline data set, and collected samples from different sources. We talk about places where people will use to collect their tickets, like kiosks. We have turnstiles where we sample from. We also went to the local trains, literally where people were hanging out of there, and we were collecting samples on the local trains as well. And I just have a timestamp here at 7:35 in the night, because this is the morning in New York. So we were still in touch with the team there and taking samples. So it was interesting to see at that time, when we generated samples from here, there was pushback on how you would process these samples. Although there was an opportunity for us to sequence them, we could not do it from within the country. So that was the reason, basically, how we collected samples. The data is still pending on for us to get that into a public domain as yet. So there was also sampling done during the COVID phase. You can see students there wearing masks, as well as protective gear and sampling. This was in the peak of, I think, phase 1 or towards the end of 2020. I just want to acknowledge collaborators here, Someshwar, whose team was also involved with that effort. So this picked up traction, and we, with Dr. Karthik Raman and Himanshu Sinha and IIT Madras, engaged with the Chennai Metro Rail, and also during the Omicron phase, we're able to collect samples. So this would actually be the second data set that's coming out from the MetaSub initiative in India. And it's actually exciting to see that. So we actually, students from their team at the Department of Biotechnology were able to collect samples from there. We've generated data. So we actually want to create the spatial relevance for that information and put it in perspective with the global data that we have. It's not about creating a negative, PR for, resistance in India here. We want to create that global picture of understanding these niches and how this resistance compares back against, right, to the environment, so that we can stitch it back to signatures that we see from animal reservoirs. It could be an opportunity where, say, hospitals from around the country, they want to come together. They want to understand what is the environmental impact of resistance buildup, right? So those kinds of questions, which we could answer using the spatial relevance, is actually becoming more and more relevant here. So from here, we have this opportunity of engaging and doing surveillance in a One Health basis using a combinatorial effect of doing metagenomic surveillance and also engaging the public. I think Dr. Rao, you mentioned engaging on the public health side, which is actually a tremendous initiative. It's the need of the hour. It needs to start at the stage of schools and go all the way up. So this is a unified way of putting into perspective all of these components, which includes international travel, medical tourism, community health tracking surveillance, some of these examples that I've mentioned in my lecture here today, talk today, and also ability to bring in a quantifiable automated way of reducing the risk of errors, human errors. Now we have AI transcending any given field whatsoever. And we have the gather to utilize some of these capabilities on empowering the metrics for which we use to do these surveillances and provide insights on how we can gather data and provide those insights. So we are so interspersed in a way that we cannot singularly look at the human host alone and provide insights on the system. We need to be accounting in for the environmental impacts of all of these domains and niches and how that triggers organisms to resist or be resilient or be redundant. So I just want to end on that note, acknowledging the people who are critical for me to be here with what's been going on in my area of research and the global impact they have created, starting with Professor Rob Knight, who's a pioneer in microbiome science and co-founder of the Earth Microbiome American Gut Project, among many others. He's now at UC San Diego. Professor Mark Hernandez, postdoc mentor. Again, I talked about his work. Peter Grofman, City University of New York. Chris Mason, Metasub Consortium. Jian Harihan was my PhD advisor at MS Swaminathan Foundation. Narasimha Rao, Karthik, Raman, Himanshu Sinha, Professor Indumati, Dr. Damodharan. Thank you so much for the invitation here. Dr. Madhimar Manikandan, and really want to acknowledge all of them, as well as finish off with this quote from Sidney Brenner. Progress in science depends on new techniques, new discoveries, and new ideas, and probably in that order. Thank you so much for your attention. Take questions.
So now it's open for questions actually. So audience, if you have any questions, you can ask, please. Any comments or questions?
Oh, thank you. It's a great talk actually. So I have only one question. When I understand that if I move from here, Pondicherry, for example, to other state, the microbiome will change. So when everything is so dynamic, how it will help in a proper decision-making system, what you are proposing?
Especially at the host level, right?
Yeah.
Yeah. Yeah, it's a good question. So again, I talked about these niches. I talked about commonalities. So when we talk about the change, the magnitude of change in orders of magnitude, right? So those are specific trigger effects, which are traceable and trackable through time. So to make these decisions, you need to have that informed guidance, which is driven by data. Yes, you know that you have a number of communities changing for somebody who's a South Asian living in the Western world. A classic example is this organism called Prevotella copri, which has now been renamed as Segatella copri because of Nicholas Segata's work. Prevotella copri was reported by a group in NYU as a biomarker for rheumatoid arthritis. And it basically raised a red flag. And they're saying that, oh, if you see an abundance of P. copri in human microbiome samples, okay, this person may be having a risk for RA. But when they were sequencing samples from, say, the Indian subcontinent or South Asian samples, we were seeing an extended spike in abundance for P. copri. And I myself got my microbiome profiled in the US. And I could see a spike in Prevotella copri that did lead me to go to do a blood test saying, oh, I want to screen myself for RA. But it turns out it is a commensal organism for a South Asian inhabitant. And you could use that as a health metric, as a metric for an optimal gut, right? So those are kinds of interesting insights that you could have with these commensal biomarkers within the microbes itself. So that is actually an interesting kind of that you could tap into.
Thank you. Thank you. Anybody have any questions? Yes.
So one very small question out of curiosity, like if we have soil and human microbial signature, can we correlate with nation specific microbial signal? Like in a nation, in one nation, there can be this signature of like microbial association.
You're asking if it can be country specific.
Yeah, we can correlate in country specific.
A country specific thing will be very challenging, but given how much cosmopolitan the whole world has become, right?
Yeah,
So
yeah.
if you think of New York City itself, right? So you have so many citizens from
Diversity.
so many countries living there,
Mhm.
different types of cuisines there, the
Mhm.
dietary, combinations that are there, people are exposed to.
Mm.
One interesting thing is you could pick up signatures associated with those dietary signatures. For
Mm.
instance, if you have foodborne microbes associated with food types.
Yeah.
So example being Korean food type called kimchi.
Mm.
Korean kimchi is abiotic, you have abiotic microbes associated with them, fermentative microbes.
Fermented there.
So you could find those signatures there.
Mm.
It becomes very challenging when you go into the nation
Space. Okay.
space,
Mm.
but there could be a gradient you could establish if you think about low middle income nations,
Yes.
right? Developing nations. You have certain biomarkers that are specific to these groups. Like
Based
the
on
Prevotella example I took, or the Segatella example I
Okay.
took. You could have other examples that you could tap in. And however, right now, my entire conversation has only been on bacteria. And mind you, having studied fungi myself, I'm a huge proponent for understanding the microbiome, which actually is another hidden signature. Although they're not as dominant as bacteria, they are having relevant niche roles and how they interact with the bacteriome or even the virome, for instance. That's another dark matter altogether. There's a lot of insights coming on that front. Maybe those could lend those insights where you're going to dissect into specific nation-centric identity. In those cases, you have these opportunities to tap into. It's again the tipping point of understanding how the data can interact with each other and also give the contextuality for it.
Okay, so
Thank you.
So one more thing is that you have mentioned that one gene from New Delhi was found in Arctic zone also. Is that it is because of the same environmental conditions back in some era, or it is due to the mobilization of
Great question. So one of the hypotheses for that was you have migratory birds
Mm.
that go from north to south, different parts of the globe, right?
Yes.
Driven by seasonality. One of the hypotheses there is you have a vector. In this case, this is the migratory movement of birds that are going towards the Arctic side, that are traversing through from South Asia potentially. Given that the hypothesis here is it originated from New Delhi, which I don't believe in. And my contention is there needs to be enough surveillance done from source to sink to blame just one city and one nation. That's not the case. That's not correct. You need to have the metrics to go from source to sink to establish that. I think that needs to happen. And also there was an opportunity during the Commonwealth Games in 2010. The first report for NDM came in 2008. Commonwealth Games happened in 2010. But there are follow-up samples collected during the time from sewage samples and water samples, which were processed, but the data was not released. So we don't know exactly at that time point what happened, but there could have been an opportunity because people from different parts of the globe were there at that same point. So those are kinds of opportunities that we could tap into and then provide the contextual relevance for the information.
Thank you, sir.
Thank you.
First of all, thank you, sir, for such wonderful presentation. I just wanted to know, like, apart from the sequencing and the 16S RNA footprinting technology, what are the other advanced technologies that are used for identification or screening of the microbes?
Yes, you can have a combination of different. For instance, there's Molly-Top that was mentioned. In fact, Dr. Damodaran has worked on these areas of, taxonogenomics. So you could have a combination of doing metabolomics. You have functional signatures associated with microbiome information. You have, again, groups such as short-chain fatty acids that you want to look at, SCFAs, some of them being propionic butyrates. You can have Molly-Top-based surveillance mechanisms for organism identification, where you're tapping into the protein profiles of the organisms when they are metabolically active. So, but also I want to mention that you have the ability to do, again, mass sequencing, doing metagenome-based assemblies, but at the same time, there's also a requirement to do culturomics. You always have to fall back to some of the classic approaches where you want to look at the phenotypic signatures for the organisms and how they cope or adjust to these HGT events, horizontal gene transfer events. It has to be a combinatorial way of doing it. Although you can only do a sparse representation in culture, but you still need to have that information to understand the phenotype of how the organism responds at the end of the day. So it's a combination of old school, I wouldn't say old school, classic techniques, but goes into the genomics and the metabolomics and the proteomics realm as well. So you have places, techniques like Raman spectroscopy, which can also be used in the metabolic sense to fingerprint and understand how these proteins differ from each other or in terms of identity. So it's a combination of all of these.
Yeah.
as a-
Thank you, sir. Thank you
Yeah.
so much.
Anybody else have any questions?
Thank you.
Thank you, sir, for the enlightening talk. And thank you, one and all. Now I request Dr. Veni Subramaniam, Assistant Professor, MGMRI, to give the final vote of thanks.
Thank you, Roja. On behalf of MGMRI, I sincerely thank our Chancellor, Shri M.K. Rajagopalan, sir, and the Vice Chancellor, Nihar Ranjan Biswasti, for the constant support for all these endeavors. And thank you, Vice President, Dr. S.R. Rao, Research, Innovation and Development. Sincere thanks to our Dean Faculty, Professor of Eminence, Ananthakrishnan, sir. And I'm privileged to thank the speaker of the day today. And thank you very much. So far, we have been thinking that we are carrying only luggages with us. We are also transferring the microbes to different parts of the country, and we are also getting them here. Thank you all. And I also thank all the constituents of Sri Balaji SBV. And I thank the hall in charge, AV, and all office staffs for their constant support. Thanks for the energy which brought us together for this day. Thank you all. Good day.